At the Max Delbrück Center in Berlin, Mina Gouti and her team are conducting fundamental research on neuromuscular diseases.
We are using human pluripotent stem cells to build advanced human models and study diseases that affect the neuromuscular system. And the neuromuscular system is the system that allows us to move. And in certain cases, this system is affected and results to severe diseases that affect the life of the patients, the quality of the life, but also the life expectancy.
These diseases include, among others, amyotrophic lateral sclerosis, ALS, or spinal muscular atrophy, SMA. SMA is one of the most common genetic conditions affecting children today. Worldwide, 1 in every 6 to 10,000 babies are born with it. 2 new ALS cases occur each year within a population of 100,000 people.
In the past, many labs have used animal models, for example, to study these diseases. But there are certain differences between mice and humans. And in order to move forward, we also now need more complex human models that allow us to understand the mechanisms of complex diseases.
During embryonic development, motor neurons form in the spinal cord and establish connections with skeletal muscles to control our movement. In neuromuscular diseases, these motor neurons die.
So a key focus of the lab is to use human pluripotent stem cells and direct their differentiation into all these different identities of spinal cord motor neurons and skeletal muscles. This will allow us to understand why certain types, certain motor neurons are affected in a disease, whereas other motor neurons are not affected.
This is achieved with the help of organoid models. The Gouti lab pioneered the first human neuromuscular organoid model to study such diseases. Organoids are tiny, self-organizing, three-dimensional tissue cultures.
So here we see in the dark part of the organoid is the muscle, and in the light part is the neural. So this culture looks pretty good. They mostly have the same kind of neural muscular compartmentalization and structure, and so this is a good sign. And these are around day 100, so they're quite mature at this stage.
These tissues, they interact, and there is a crosstalk that also allows them to mature. Our model is really good to study these diseases because it's complex enough so we can try and understand which cell type is affected first in the course of the disease. What was really striking is that they also start talking to each other, and the spinal cord motor neurons innervate the muscle and instruct the contraction of this muscle as it happens in the human body.
Muscle contraction is even visible when looked at under the microscope. However, they don't completely represent the human physiology just yet.
There are still things missing from these models, for example, vascularization. We generate organoids from many different tissues. We even generate complex organoids now that we can have more than one tissue in one organoid. But still there is no vascularization. There is no blood flow inside these organoids as we have in the human body, right? And this is really important because in order to keep these models for very long in culture and to make them mature, you need also the support from the blood circulation.
The organoids can be kept alive for months when nurtured. In the future, the goal is to have them reach adult age.
The beauty of it is that we can now start asking how the other cell types affect the motor neurons. What is causing the motor neuron death? And we have certain cases where we see that other cell types are affected before the motor neurons.
To examine the organoid models, they are fixed in place, then frozen and cut into wafer-thin slices. To make the microscopic structures visible, these samples are stained with different markers that can then be examined under a fluorescent microscope.
So we can see the muscle in magenta and the neuromuscular junctions in cyan.
Neuromuscular junctions are the chemical synapses between a motor neuron and a muscle fiber. Improper function of these junctions can lead to the development of progressive neuromuscular diseases.
A big challenge in the organoid field right now is reproducibility and scalability. One approach that my lab is taking now is to also use liquid handling robots. We try to establish this automated pipeline now, not only to maintain routinely the stem cells in culture, but also to be able to generate organoids using the liquid handling robot. This will allow us to generate like thousands of organoids in 1 hour. Can have more statistical power in all our experiments because we can do them in much larger scale using these machines that was not possible before. So if we want to test certain drugs, for example, if they will have an effect in humans, we can have now an intermediate step. Instead of going directly from the mouse models to the patient, now we can have these organoid models, the human-specific models where we can test the effect of the drug. And of course, this will also contribute to reduce the number of animals that we use in research, because we can instead use the in vitro models.
The organoids are then used for drug screening on a larger scale.
To see which drugs enhance the survival of the muscle or the motor neuron, so which one could be potentially go then into clinical trials. When we do drug screening, we look at the effect of the drug on the number and the size of the neuromuscular junctions.
Focus for the future is to use these organoid models and make patient-specific organoids so that we can do high-throughput drug screens and identify novel drugs that could be used to cure diseases. The challenge here is that, in order to cure complex diseases, you might need also complex treatments. And I think this is exactly the power now of this model, that we can test the combinations of different drugs and also see which drugs are effective to specific patients.